Okay, question 2 then from the 2023 Higher Human Biology past paper. The diagram represents stages of an enzyme catalyzed reaction. So you've got your substrate, your enzyme, your active site, you've got stage 1 turning stage 2, and you've got some products being produced at the end. You're being asked to use the diagram to explain why this reaction can be described as catabolic. So let's remind ourselves what the meaning of those words are. If something is catabolic, it means it's a breakdown reaction. If something is anabolic, it's a build-up reaction. Okay, so I always remember that as like if you're taking anabolic steroids, you're getting massive, you're getting huge, you're trying to build up muscle. So anabolic is build up, which means catabolic has to be a breakdown reaction. So basically, your question is, how do you know this is a breakdown reaction? Well, we started off with a larger substrate and ended up with smaller products. So it's a breakdown. If we had started with a small substrate and ended with a big product, that would be a build-up reaction, an anabolic reaction. So, how can we tell that this reaction is a catabolic reaction? The way we can tell is because we have large substrate being broken down into smaller products, and that's how we know it's a catabolic reaction. In 2ii, the diagram shows induced fit occurring between the enzyme and its substrate molecule. Describe what happens during induced fit. So this is all pulling on our knowledge of enzymes and substrates. So we know enzymes or substrates have to be specific to one another. Because every enzyme has its specific substrate. Enzymes can't just break down whatever they feel like. Although you can have something that is specific, so you got an enzyme that's specific to the substrate, they need to have something called an induced fit. So there needs to be something called an induced fit. And what that means is the substrate and the enzyme, even though they've come together, they need to mold in together to get a nice tight fit around each other. I always think of it as if you're giving someone a hug, you can put your arms out and they can put their arms out to come and hug you. But when they come close, you've got to actually wrap your arms around them, right? So you've got to actually like move around a little bit to get a tighter fit. So that's basically what's happening during induced fit. The thing is, though, the substrate's not what's changing. So what's going to be changing during induced fit? It's the active site. So the active site is going to change shape to better fit the substrate. So that's what induced fit is all about. It's almost like that active site's trying to give the substrate a hug. Once the reaction is complete, the products are released from the active site and the enzyme can be reused. Explain why the products leave the active site. So that's a really good question for us to look at. Let's look at that diagram again. So we can see from this diagram, the substrate and the enzyme's active site are going to be specific to another. And we use a word called affinity to describe that. What we say is the substrate and the active site have a high affinity for each other, meaning they're highly attracted to each other. They want to be in contact with each other, and that's why you get enzyme substrate binding and induced fit taking place. However, once you've produced products, the products aren't bothered about the active site. They don't care. So they want to leave the active site because they have a low affinity. So because they don't have an attraction to the active site, they have no reason to be there. And that's why your enzyme releases them. So how can we translate that to an answer? We could say products have a low affinity for the active site, or we could say that products no longer fit the active site because it was the substrate that was specific to the active site, not the product. Question 2b, Parkinson's disease is caused by low levels of dopamine in the brain, resulting in poor muscle coordination. And Parkinson's disease can be treated using the drug L-DOPA, which is converted to dopamine in the brain. L-DOPA is produced commercially in a reaction vessel using the enzyme tyrosinase. I always struggle with these names, as shown. Now, whenever you're doing an enzyme question that has something to do with an in-context example, take a second to identify what is your substrate in this question, what is your enzyme, and what is your product. So if we're to look at that for a wee second, this here, tyrosine, that would be the substrate. Tyrosinase is going to be the enzyme. L-DOPA will be your product. It's helpful to do that because then if anything changes with those levels, you're able to basically understand what's going on. So the graph here shows the results of using different concentrations of the substrate, tyrosine, and the concentration of L-DOPA produced in the reaction vessel. So what we can basically see happening in this graph is we see there's a pretty steady increase, and as we're increasing the tyrosine concentration of L-DOPA is increasing. Then we hit this point at around 5 milligrams per centimeter cubed. We hit a plateau essentially, and it continues on that kind of plateau. Using the information from the graph suggests why a tyrosine concentration of 5 is used in the commercial production of L-DOPA. Well, it's commercial. You don't want to waste your money producing this drug. So the reason we use 5 is it's the lowest concentration of tyrosine that produces the highest amount of L-DOPA. You don't, if you use 8 milligrams of tyrosine, you still produce the same amount of L-DOPA. So what's the point of using 8 when you can use 5? For this question, you can put in the values if you wanted to. You could say, 5 milligrams per centimeter cubed is the lowest concentration that produces 0.01 milligrams per centimeter cubed of levodopa, and that's totally fine as well. It's quite good to put in numbers sometimes when you're dealing with a graph, but if you just described it in words as well, you would still get the mark for that also. And the final question here, many enzymes are used in commercial processes. When using enzymes in these processes, the end product is regularly removed. Suggest why end products are removed during production processes. To understand this, let's take a look at a little diagram here. So here we've got a metabolic pathway of some description. We've got the start of the pathway, some intermediate molecules, and the final product. Now, the reason that we have feedback inhibition in our bodies or in nature is so that we don't waste resources and we maintain levels of a certain compound at an optimum level and doesn't get too high or too low. So what's going to happen is you're going to start producing these intermediates, and as you go through this process, you're going to produce a product, and that product's going to build up. The purpose of feedback inhibition is that the build of that product is going to inhibit one of the earlier enzymes in the process. Why is that important? It's to stop the overproduction of that product. Okay, so that's what's going to happen, like in our bodies, for example. However, in a commercial context, you actually want there to be as much end product as possible. This is what this question is talking about, the commercial context. So here, you're actually going to want to remove the product so that you don't get any feedback inhibition, okay? So if you don't have as much product, you're going to get less feedback inhibition, and that process is going to keep working and working and working. It's not going to get slowed down. So therefore, the answer to this question is going to be to prevent end product inhibition. Okay, so if you remove that end product, you're removing end product inhibition, or feedback inhibition is another way you could say that.